Now, once the cells have formed the inner cell mass, if we now look at our little blastocyst here again, you can see that of the trophoblast cells, some of them are in contact with the inner cell mass and some of them are not. So the next step will be that the ones in contact with the inner cell mass will specialize in a different way from those that are not in contact. Similarly, if you look at the inner cell mass cells, some of them are exposed to the space in the center, to the coelom, to the opening in the middle of the blastocyst, and some of them are in contact with trophoblast cells, and again, these will have different fates. So we went from one cell kind to two cell kinds. Those two cell kinds now generate four cell kinds, and we're looking at a cascade of differentiation following from these developmental signals which are transmitted from cell to cell. So different fates come as a result of different positions, and although we do not fully understand the nature of all those signals, we are understanding some of them, and this is a work very much in progress. So here you can see in the diagram, trophoblast cells not in contact with the inner cell mass will follow a different fate from those that are in contact. Inner cell mass cells exposed to the blastocele will follow a different fate from those that are not exposed to the blastocele. The idea behind this is called positional information by Professor Lewis Walpert. And a good way to help understand this is to look at the development of the fruit fly. Now, at first you might think fruit flies are a long way from human beings. Even in this idealized view, it does not look much like a person. But what we now know is that the developmental signals and the developmental genes which help express different parts of the embryo during the course of development are actually common to all animals, and indeed common to animals and plants in many cases as well. It's as if there is one set of developmental genes which are activated at different times in the development of all living things. And it would be fair to say that this was a very unexpected finding indeed. And this is despite the fact that the different parts of an organism can be very varied between different species. So for instance, the eye of a fly is very different from the eye of a human, but the underlying genes that say make an eye turn out to be almost identical. Same would be true of heart formation, for instance, aspects of brain formation, aspects of the genital apparatus, and aspects of the limbs. Despite the fact that they vary from animal to animal, the developmental signal that says form a limb turns out to be very similar. And if you want to know more about this, then Hox genes would be a good starting place to gather more information. But I can give you one example. There is a mutation found in fruit flies called eyeless, and what that does is to affect eye development, as you might have guessed. And because it's easy to breed fruit flies and to analyze their genetics, we can identify the gene that's involved in this, the eyeless gene. The particular gene involved is known as PAX6. And if you were to look at the human genome, you would find the gene very similar to PAX6 in its structure. And from time to time, children are born with eye defects. A common one is a reduction in the size of the eye. And because we know the nature of PAX6 in fruit flies, we can explore the human genome for a corresponding gene, and we find one. And what we know is that if we were to remove that gene from a human and to place it in a developing fly, it would restore eye formation in that fly. Now, you might be wondering, would that extra eye be a human eye or a fly eye? And perhaps disappointingly, it's a fly eye. The signal coming from PAX gene tells the fly to make an eye, but the fly interprets that signal in the light of its evolutionary history, and therefore it makes a fly eye. If you could do the converse experiment, take that gene from a fruit fly and implant it into a human, it would be illegal, but technically it would be possible, then an eye created by that gene would be a human eye and not a fly eye. And this gives us a very good way of looking for the genes that underlie developmental defects in humans. If we found, for instance, a heart defect in humans, we could rapidly scan the fruit fly to find a gene that caused heart mutations in the fly, and then check to see if that gene was altered or abnormal in the human that was suffering from that heart condition. Again, if you want to know more about this, PAX genes would be the thing to look up.